Today we'll be discussing the first part of the Genodermatoses lecture, which comprises ichthyosis and epidermolysis bullosa. Well, first we have to know the difference between two terminologies, genodermatoses and congenital diseases. Genodermatoses is a genetic dermatological disease. But what about congenital dermatosis or congenital disease? The word congenital simply means that this disease is present at birth. So, congenital disease could be a genetic disease and could also be not a genetic disease. For example, congenital syphilis. It's an infection that could be contracted either during pregnancy or during delivery. What we will be discussing is the genetic dermatological disease, a disease due to a genetic abnormality. This abnormality could be either in a single gene, multiple genes, for example, atopic dermatitis, or it could be chromosomal, for example, Down syndrome, where we have an extra chromosome. We've already explained that. Now, we'll be discussing the ichthyosis group, and yes, it's a group. There are multiple subtypes, but we will be discussing only the three main subtypes, which are ichthyosis vulgaris, X-linked ichthyosis, and lamellar ichthyosis. Well, first of all, what is the main clinical picture in ichthyosis? It is the scale. And as long as the scale is the important clinical picture, it should be well described. Description is the same in the three subtypes. These are polygonal scales, attached at the center and free at the periphery. You'll be noticing that there are only some variations in the distribution of the scales, the color of the scales, and the size in the three subtypes, but they are all polygonal scales, attached at the center and free at the periphery. But why does ichthyosis happen in the first place? Now, this is a very important diagram. If you observe, this is the stratum corneum. It is composed of cells, the corneocytes, embedded in a matrix. We usually describe this appearance as a brick and mortar appearance, as if the cells are bricks embedded in a cement material. Now, this relation, the cells and the cement material, is important. Why? Because it allows for the normal desquamation of the cells. We all know that the epidermis is composed of layers: the basal cell layer, the spinous layer, the granular layer, and the uppermost layer, the stratum corneum. What normally happens is that the basal cell layers, the stem cells, differentiate and proliferate, giving the spinous cell layer, which further differentiates to the next layer and further differentiation to the stratum corneum that finally desquamates. If it happens that the stratum corneum cells cannot desquamate, what do we expect? We expect that more layers are formed with the differentiation of the epidermis, while the upper layers cannot desquamate, and therefore there will be thickening of this upper layer, giving hyperkeratosis, which is the abnormality in all types of ichthyosis: impaired desquamation, retention hyperkeratosis. Now, if you look here, the cells are rich in a protein called filaggrin. The cell envelope is also enriched in filaggrin, which interacts with the lipid cement material. Now, this cement material is mainly composed of lipids like ceramides, cholesterol, and free fatty acids in fixed proportions. Any variation in this normal structure will lead to impaired desquamation and ichthyosis. This is what we've been saying. This is the normal stratum corneum with the corneocytes embedded in the lipid matrix, which is reminiscent of the brick and mortar appearance. Ichthyosis vulgaris is the most common type, as the name implies, vulgaris, which means the most common type. It's an autosomal dominant disorder, and the defect is in the filaggrin protein we've just mentioned, defect in the filaggrin gene. Now, besides being an important composition in the cell envelope and inside the corneocytes, filaggrin, when it degrades, it gives us important degradation products, of which is the natural moisturizing factors. As the name implies, natural moisturizing factors are important for moisturization. So if I have a defect in the filaggrin gene, I expect impaired relation between the stratum corneum cells, the corneocytes, and the lipid matrix, leading to impaired desquamation. And I also expect a decrease in the natural moisturizing factors, leading to dryness. Now, you'll be taking the atopic dermatitis lecture, and you'll be noticing that atopic dermatitis, besides being due to environmental factors, is also due to multiple genetic defects. Of these genetic defects is a defect again in the filaggrin protein. So we expect that ichthyosis vulgaris will be having the clinical picture of ichthyosis plus associated features of the other disease with the same genetic defect, which is atopic dermatitis. We might find manifestations of atopic dermatitis in ichthyosis vulgaris patients. This is the diagram that shows how the profilaggrin, the precursor of filaggrin, is converted to filaggrin, and finally to the NMF, the natural moisturizing factors. What about the clinical picture? Ichthyosis vulgaris starts after 3 months of age. The baby is born normal. Manifestations start to appear after 3 months. The scales, again, are the polygonal scales attached to the center, free the periphery. They are coarser over the extremities than over the trunk. They're fine scales. And characteristically, a very important differentiating point is that they spare the flexures, the major body folds. For example, the popliteal fossa and the antecubital fossa. Also, you'll be observing, or the patient's mother, for example, will be saying that the child improves somehow during summer and worsens in winter. Why is this? Because dryness normally increases in winter, even in normal subjects. And in summer, the sweating might make the condition appear less severe. As we've mentioned, atopic dermatitis manifestations might be found, mostly palmoplantar hyperkeratosis and keratosis pilaris. If we take a biopsy, unsurprisingly, we will be finding the main defect, which is hyperkeratosis. Regarding the prognosis, this subtype usually improves somehow with age. This is a limb showing the polygonal scales attached at the center and the free of the periphery. And characteristic here is the sparing of the popliteal fossa. This hand here shows some mild hyperkeratosis due to associated atopic dermatitis manifestations. Again, the biopsy will be showing hyperkeratosis. The next subtype is the X-linked ichthyosis, and as the name implies, it's an X-linked disorder. Actually, it is X-linked recessive. So, do you expect this disease to be marked or to be manifested in males with a single X chromosome or in females who have 2 X chromosomes that might compensate for a defect on one? Of course, in males. The defect in this disorder is in an enzyme called steroid sulfatase, and as the name implies, it's an enzyme that acts on cholesterol sulfate, converting it to cholesterol. Now, if there's an abnormality in the normal fixed proportion of ceramides, cholesterol, and fatty acids in the lipid matrix, we expect again impaired desquamation of the corneocytes. What about the clinical picture? Now, ichthyosis vulgaris, if you remember, starts after 3 months of age. This subtype starts before 3 months of age. Scales were fine in the ichthyosis vulgaris. Now here they're tan, dark tan or grayish. Scales characteristically spare the flexures in ichthyosis vulgaris, but in this subtype, it characteristically involves the flexures. The scales markedly affect the major folds, especially the neck and the lateral face, giving a very characteristic appearance, what we call the dirty neck or unwashed look. This is a very important differentiating point, by the way. If you look here, you'll be finding the dark tan scales that are coarse over the trunk. Now, again, symptoms improve in summer, worsen in winter, but there is no improvement with age, unlike ichthyosis vulgaris, and we do not have associated atopic dermatitis manifestations. These are the scales involving the neck and the lateral face, as you can see, and again, the biopsy will definitely show hyperkeratosis. The last type, the lamellar ichthyosis, which is the most severe type, is either apparent soon after birth Or the baby might be born in a collodion membrane. Later this membrane dries up, cracks, and exfoliates, revealing the manifestations of lamellar ichthyosis underneath. Now, this is generalized, involving the entire body surface. The scales here, again, are polygonal, attached at the center and free at the periphery, but these are large plate quadrilateral-like scales that may reach up to 1 centimeter in size. The entire body surface area is involved. The palms and soles show hyperkeratosis. The face always shows ectropion, almost always. This is a characteristic feature. And if we take a biopsy, definitely hyperkeratosis will be observed. So how do we treat these cases? It's a genetic disease. All we have currently are symptomatic treatments, some applying topical moisturizers, emollients, topical keratolytic agents, and it's always useful to apply the emollients over moist skin as if to form an occlusive layer that will occlude this moisture on the skin. And of course, not to forget the keratolytic agents, but these have to be applied only to localized areas because extensive use over large surface areas always entails the risk of systemic absorption and side effects. In severe cases such as lamellar ichthyosis, we use systemic treatments, which is the systemic retinoids. In this case, it's the acitretin. The next half of the lecture is Genodermatopigmentosum. Again, it's a genetic skin disease, a genodermatosis, but in this case, the main problem is extreme photosensitivity. It's a photosensitivity to ultraviolet radiation. Normally, we're all exposed to ultraviolet light. Ultraviolet light normally can cause damage to our DNA inside our cells. And if this damage is left unchecked, cells divide, the damage increases. We always have the risk of mutation and cancer. But why doesn't this happen to most of us? Because we have checkpoints for our cell cycle that detect the damage, that there's a damage in the DNA, therefore they stop the cell cycle, repair the DNA damage, and then allow the cells to divide again. This is the problem in patients with xeroderma They have defective DNA repair. After the ultraviolet rays induce damage in the DNA, they cannot repair their DNA due to defective DNA repair enzyme, defective DNA endonuclease. Now, this is an autosomal recessive disorder. I think by now you must know that the most important clinical picture is photosensitivity that starts early in age, from trivial sun exposure reaching to sunburn. Then the patient or the baby starts to develop freckles. Later on, the patient develops mottled hypopigmentation, so now they have mottled hyperpigmentation and hypopigmentation. Then the patient starts to develop pre-malignant skin changes and finally malignancy. The most common subtypes is basal cell carcinoma, squamous cell carcinoma, and less commonly malignant melanoma. This defect in DNA repair is not only present in the skin, but all over the cells of the body, so we expect the patients to have internal neoplasms as well, particularly in the central nervous system. This is a picture of a baby developing sunburn. After sun exposure, you can see the sunburn has affected all sun-exposed areas. Another child who started to develop freckles, then mottled hypopigmentation, pre-malignant skin changes, and the rodent ulcer or basal cell carcinoma, quite clear in this. Now, the other system or the other main organ that is affected is the eye. And as expected, the anterior segment of the eye has more severe affection than the posterior segment due to ultraviolet rays involvement, starting from the lids, moving on to the conjunctiva, the cornea, and the lens. Patient will have ectropion, conjunctivitis, corneal opacities and cataract, which you can see here. The ectropion is evident, the corneal opacities, and the malignant eye changes. Of course, we do not forget that neurological manifestations are common, with progressive neural degeneration in 20% of cases, and of course the CNS neoplasms. How do we diagnose? We should suspect this case if photosensitivity, sunburn starts at a very, very early age, find freckles at an early age, and of course skin malignancy at an early age. age. The most important part of the management is protection from sun exposure, of course by protective clothing, long sleeves, trousers, wide-brimmed hats, sunglasses, using ultraviolet film protective layer for the windows, and of course the regular frequent application of sunscreens. There are some papers that advocate the use of systemic retinoids to decrease the incidence of malignancies in these patients. Thank you.